All right, good morning, everybody, and welcome to today's class. Okay, good morning, like I said earlier. So welcome to today's class. Last time, we looked at the nucleic acids. Please don't drop anything here, please. Uche, Angel, I will kick you out. Take your discussion to your class groups. All right. Last time we looked at nucleic acids. We said there are basically two types of nucleic acids, the ribonucleic acid known as RNA and the oxyribonucleic acid. And I said the major difference between, although there are other differences, but structurally, what distinguishes a DNA from an RNA is the presence of oxygen in addition to the hydrogen atom at carbon position 2 of the pentose sugar, whereas in DNA, the oxygen is missing at that position. And that is where we say it is deoxy, that is, an oxygen atom has been removed in that particular position. That is carbon 2 of the pentose sugar. That is deoxyribonucleic acid, and RNA is ribonucleic acid. That is, RNA as a ribose sugar and DNA as a deoxyribose sugar. We also said that the carbon of the sugar is always labeled with prime to distinguish them from the carbon of the nitrogenous bases. And we looked at the structure of the DNA and RNA. In both, the base is always attached to carbon 1, and hydroxy group to carbon 3, and the phosphate group to carbon 5. Carbon 2 is what distinguishes them, like I said earlier, where you can either have an OH or H, depending on whether you're looking at DNA or RNA. Then we looked at the nitrogenous bases. We said there are basically two major types of or classes of nitrogenous bases. You can have purines or pyrimidines. We said the purines are double ring and they are A and G, that is adenine and guanine. We said the pyrimidines are single ring and they are cytosine, thymine in DNA and uracil in RNA. We look at the structure of the DNA as elucidated by Watson and Crick, and we looked at people that contributed to the structure of the DNA. We looked at Erwin Chargaff, and Rosalind Franklin. And I remember giving you an assignment to look at the contribution of other scientists. Then we look at the pairing of the nucleotide bases in these nucleic acids. We said pyrimidine pairs with adenines. Sorry, pyrimidines pairs with purines, and purines pairs with pyrimidines. That is, A pair to T, G pairs to or with C. A and T with double bond, C and G, or G and C with a triple hydrogen bond. Then we look here further to see how the nucleotides, how they pair. We said one DNA is antiparallel in that one runs from 5' to 3', the other one from 3' to 5'. We said the backbone, which is the phosphate, known as the backbone, linked with the sugar with a covalent bond, while the bases are joined by hydrogen bond. And we said, how do we know which one is 5' to 3', which one is 3' to 5'? We look at what's it called? We look at the sugar, the sugar, and see the carbon that is exposed, right? So that carbon determines whether it is 3' or the 5'. And this shows the structure of how phosphodiester bond forms between the nucleotides. We said the incoming nucleotide will always pair with a 3' carbon. So that 3' carbon is always exposed, right? You need it exposed, you need it available, so that the incoming carbon, the incoming nucleotide can attach it with the 5' carbon that is carrying the phosphate group. That is why you have phosphodiester bond formed between nucleotides. And today, without much ado, let's go to the business of the day. Today, we are going to start a class on central dogma, where we are going to look at how information is transmitted from DNA to RNA to protein. Now, the central dogma holds genetic information that is expressed in a specific order from DNA to protein synthesis, right? Like I said earlier, the cell doesn't read DNA, right? What it understands is protein. The language spoken, understood by the cell is protein, right? So there is transmission of information from the genetic elements, DNA, to RNA, to protein. And the type of RNA we're looking at here is mRNA, that is messenger because there are different classes of mRNA, as we shall see later in the course of this lecture. So the central dogma simply talks about transmission of information from DNA. You know that information is encoded in the DNA, right? So it has to be interpreted. And I remember saying maybe somebody gave you Greek letters, right? A letter that's written in Greek language. Then you need somebody to interpret it for you. That is what the cell is doing here. So prior to, so we have DNA to RNA to protein. But before DNA can change to RNA, there is what we know as DNA replication. The DNA replication is a process whereby the cell make more of its copies, right? The cell is making, like you are making a photocopy. That is what is happening at DNA replication. The DNA is making more copies of itself changed or being transcribed to messenger RNA, right? So first we have DNA replication, whereby the cell is making more of its copies using itself like as templates, right? And we're going to see how that takes place soon. Then after replication, and this replication takes place or occurs during the cell cycle, during interphase, right? Then the stage of the interphase is synthesis. From your secondary school biology, you were taught cell division, right? You have interphase, metaphase. Sorry, you have interphase and mitosis and what have you, right? So interphase is divided further into three, right? You have the G1 phase, you have the S phase, and the G2 phase. So that phase where DNA replication occurs is the S phase of the interphase. That is S for synthesis. So what is actually being synthesized there is DNA, right? In the process known as DNA replication or DNA duplication, where the cell make more copies of its DNA content, right? So after replication, we have, that is DNA to DNA, DNA making more copies of itself, like you have DNA making more copies of DNA. So DNA to DNA is known as a replication. After that process, you have transcription. Transcription is a process whereby DNA is transcribed to mRNA or changed to mRNA. Don't forget that DNA is double-stranded and RNA is a single-stranded. And as you can see, this is a double strand. This is one strand. This is another strand. But here you have your RNA that is just one strand, right? So that is a DNA transcription. We are still going to look at all of these stages one after the other. Then after RNA is transcribed from DNA, you have you have protein synthesis, right? Through a process known as a translation. So DNA to DNA replication, DNA to RNA transcription, RNA to protein translation. Okay. So let's look at DNA replication. DNA replication is the process of making copies of DNA, like I said earlier, like you are making photocopies. Maybe you have just a copy of your notes, and you want to give some of your friends not come for class. So you want them to have the notes. You go and make copies and give it to them. So that is just what DNA replication is. And we said that stage of the cell cycle where DNA replication occurs is the S phase, right, of interphase. And this shows the cell cycle here. You have the interphase taking the larger portion of the cell cycle. Then from there to mitosis. Mitosis to prophase is further divided into prophase, metaphase, anaphase, and telophase, which is known as the M phase together. Then you have cytokinesis. Cytokinesis simply means division of the cytoplasm. You are still going to do all of this later, right? So that is DNA replication. Simply put, DNA replication is a process whereby the cell make more copies of its DNA content or duplicates or synthesizes its DNA content, right? So you saw that DNA is double-stranded like this. DNA is double-stranded such that this is double strands of DNA. We said that DNA one runs from 3' to 5', the other one runs from 5' to 3'. So each of the strands serves as templates for DNA replication, right? Each of the strands. So during DNA replication, the two strands will separate. We are going to look at that. The two strands will separate so that this one on its own will make its own complementary strand. This will also make its own complementary strand. So at the end of the process, if you have 4, right? Eventually, you are going to have 4. You are going to have 8 because each of these will go on to make copies of their DNA or to duplicate themselves. Okay. Then, now, methods of DNA replication. There are 3 models that have been proposed to explain how this DNA replication actually takes place. The first one is the semi-conservative method. And what this model says is that each daughter cells has one parental strand and a new strand, right? As displayed here, right? This is the parent cell or parental strand, rather. This is the parental strand. So the semi-conservative model is saying that DNA replication, what we have eventually is that there is one parental strand and the other a daughter cell that are formed as a result of DNA replication. Another model is conservative model, and that simply means that the parental strand is conserved, right? So the two parental strands come together and you have the new daughter strands coming together. Don't forget, I said during the DNA replication, the two strands of the DNA will separate, right? Don't forget they are joined together by nucleotides. So you have, if you have A here, you have T as complementary strand. If you have G here, you will have C. If you have T, you will have A. If you have C over there, you will have G as a complementary strand. This is how DNA is double-stranded together, and they are joined by A and T by double bond, C and G by triple bond, and A and T and A by double bond. So during DNA replication, the two of them will separate, right? Like they will divide into 2. They will be separated such a way that this one will serve as a template to make a new daughter strand. This one will serve another template to make another daughter strand. Now, what the conservative model is telling us is that during that process, eventually, when the process is completed, you are going to be having the parental strands conserved, staying together, and the new daughter strands that are highlighted or shown in red here will stay together. Whereas the semi-conservative, you say that is not totally conserved, that is semi-conserved, that one is made up of the old strand, the other one, the new strand, like that you have old strand and new strands coming together. Then when this one breaks up to make new DNA, to synthesize another DNA or to replicate itself, they will now become, this one will now become the parental strand, that they will still, it goes like that you also have a new strand and an old strand, a new strand and an old strand. Then the third model is the dispersive model, which says that the DNA is fragmented, right? Then both new and old strand coexist in the same strand. And as you can see there, this is a dispersive, like the DNA is broken down into smaller pieces. You now have them dispersed along the line in the new daughter cells or new strands, in the new strands that you make. And these are the 3 models: semi-conservative, the parental strand and the new strand stay side by side together. The conservative said that no, the parental strands are conserved, they stay together, and the daughter cells stay on their own. Whereas the dispersive model says that the DNA is fragmented and you have them together, both new and old coexist in the same strand. Now, these are 3 models that were propounded for DNA replication. So which one do we agree with as the model that is used by DNA for replication? It is a semi-conservative model, right? So the DNA replication follows the semi-conservative model where the parent strands and the parental strand and the new strands stay together, as seen here in the middle. This is the model that the DNA replication follows, whereby each new double helix of the DNA, one strand of the parent and the other one is the daughter cell. This figure shows the different replication models, right? This one is the conservative model where you have your parent cell having this. Then the first generation, this is what was proposed. Then when each of these also goes into their replication, it says this is what will be formed, right? But the experiment of please drop this, Emanuela, drop this your summary into your class group, not in my chat room, please. Right? We can't have two teachers at a time in the class. Or you let me post, you have to take over. If you have any, don't. Okay, please. Okay, so these two individuals, Meselson and Stahl, carried out an experiment to show, to elucidate which model does a DNA replication follow. And what they did was to culture bacteria in heavy nitrogen. You know, we have isotopes, right? The isotope. I'm going to remove this individual, Ademuyiwa Daniel, that you are asking me now is attendance being taken, as if you are the one that is taking my attendance for me. Go and join YouTube and go and ask them on YouTube. All right. So we have Meselson and Stahl carried out their experiments to determine which replication model does DNA follow, right? And what they did was to start the experiment by first culturing bacteria in a medium containing heavy nitrogen. That is the 15th isotope. You know, we have your chemistry, you did isotopes, right? So 15 isotope 15, right, of nitrogen, right? Then we have, after that, they transferred, after culturing it in heavy nitrogen, they transferred the same bacteria to light nitrogen, right? Then they centrifuge for 20 minutes for the first generation. Then this is what they got for the first generation. They saw that it doesn't follow the conservative. They use that to disperse. At first generation, they saw that there were two sets of bacteria, one with the heavy nitrogen, the other one with the light nitrogen, as depicted here. So they erased that model. Then proceeding further to second generation, that is after 20 minutes, they centrifuge again and observe their results. And they observed that they used that to eliminate the dispersive model. Then they were able to prove that DNA replication model actually follows the semi-conservative model, that DNA replication is semi-conservative model, that is by culturing bacteria in heavy and light nitrogen. That was how they were able to determine which model the DNA replication actually follows. So, okay, as an assignment for you, We are going to describe the Meselson-Stahl experiment in details and what will be the outcome if the experiment continued for 3 generations. Don't forget, the experiment stopped at they stopped at second generation, right? Second generation there. So if that experiment continued longer, for a longer period, what would their results actually be? So that is the assignment, another assignment for you there. Read it up and make a note and put them down in your notes. You are not submitting to me. Where you will submit, you will submit. All right. So let's look at DNA replication and how DNA replication occurs. First, let's look at the enzymes that are involved. The first enzyme is helicase. Don't forget we said that DNA is double-stranded. So for DNA replication to take place, the DNA Okay, can you all hear me? I'm trying to, I got kicked out again. I don't know why. So I don't have enough capacity here, but I'm trying to go back to share the link because it's just 270. So I don't, okay, thank you. Fifty-eight, 48, 55. 58, 48, 55. Okay, I've sent the passcode. Okay, so please let's continue. So we are here on a DNA replication. Looking at the DNA replication enzymes. We said DNA helicase helps to open up the DNA strands, right? This DNA is double-stranded, right? So it needs to be unwound or unzipped so that DNA, the replication can occur. So you need to the DNA to be opened up, right? For DNA replication to take place. So let's depict that with so you need the DNA to be opened up this way. These are the two strands. Let's say this one, okay, so you need it to be opened up this way so that replication can take place. Then as the DNA strands open up, right, there is an enzyme that prevents them from reannealing or from rejoining or from closing up again. And that enzyme is known as a topoisomerase or DNA gyrase. Sorry, no, single-stranded binding proteins, SSBDs. The single-stranded binding protease prevents the DNA from joining up by binding on the opened strands. So let's depict this circle with our SSBDs, that is single-stranded binding proteins. They are enzymes that bind on the opened strand so that they don't close up again. Then we have topoisomerases or DNA gyrase. DNA gyrase prevents torsion from forming as the DNA unwinds, right? So it is possible, like if you have your rope, right? If you have your rope, as you are opening it, it's possible that they form knots, right? So the DNA topoisomerases or DNA gyrase prevents that from forming. No, we said that DNA are wound around each other. The two strands are wound around each other. So if you are opening them up, this one going this way, this one going this way, it is possible that they form knots that prevents them from further opening up. But there is an enzyme that prevents that. You call that torsion, Torsion. There's an enzyme that prevents them from joining up. You call that enzyme DNA gyrase or the topoisomerases. Then we have a primase, RNA primase. This synthesizes primers at the 5' end of the leading strand and 5' end of Okazaki fragments of the lagging strand. The leading and the lagging strands will be seen later. So what happens is that before DNA replication occurs, let's assume that this DNA has opened up, right? So you have maybe this strand is A, C, C, A, C, G. On the template strand, you are going to have C, G, G, T, G, C, right? Now, each of them will now serve as templates for DNA replication. So this one will form its own DNA strand. This one will form its own DNA strand. So we have a T here, all right? So before that occurs, we said the DNA strand runs antiparallels. This one runs from 3' to 5'. This one runs from 5' to 3'. Now, there's an enzyme known as RNA primase that lays RNA primers, short primers about 8 to 9 bases that lays them here, that starts them. So it lays about 8 to 9 bases, then DNA can start coming before the replication can begin, right? Like like when you prime something, like you initiate it, right? So that primer is known as that enzyme is known as a RNA primase that lays short primers, RNA primers for synthesis to begin. Then we have DNA polymerase 3. DNA polymerase 3 is the one that is the enzyme that adds more nucleotide, right? DNA polymerase 3 is the enzyme that adds more the nucleotides that synthesizes them. So for A, it will bring T, for T, it brings A, for G, it brings C, and that's how the strand is copied entirely. So using parental strand as a template, DNA polymerase 3 synthesizes new DNA strand by adding nucleotides to an RNA primer or a pre-existing DNA strand. We said, fourthly, RNA primase needs to come and synthesize RNA primers, right? I said when these strands open up, the two strands open up, before DNA polymerase 3 comes in to bring the nucleotides, right? An enzyme known as RNA primase which synthesize short primers, right? On the leading and the lagging strand. Leading and lagging strand will be explained later, right? So we synthesize short RNA primer before DNA polymerization actually takes place. So after that, the DNA polymerase 3 will now begin to bring specific nucleotide. For instance, if you have A here, the DNA polymerase 3 will bring a T. If you have T here, DNA polymerase 3 will bring an A. If you have G here, it brings C. But before this occurs, you have an enzyme known as RNA primase. This enzyme will initiate the synthesis, right? Then after that, DNA polymerase 3 can now come in and do that process. It's just like when you want to pump your water and your pumping machine doesn't start until you first pour water inside it, then it now picks up, right? That is how it works, right? Or your carburetor, doesn't work, or what is that engine that pumps fuel? Your car doesn't pump fuel very well. So you need to put petrol in your mouth, like all these mechanics do. You need to put petrol in your mouth and blow it on your right before the car will now start kicking and start pumping fuel. That is how, well, that's what RNA primase does. RNA primase needs to prime that replication, needs to initiate. So it lays short primers, RNA primers, right? Before DNA polymerase 3 will now come in and bring nucleotides and elongate that strand. Then we have DNA polymerase 1. Polymerase 1 removes nucleotide of primers and replaces them with DNA nucleotides added to 3' end of the agent. So DNA polymerase 3, so DNA polymerase 1 will now come in and remove the RNA primers that is laid initially for the reaction to begin, and now add the respective nucleotide. DNA ligase. DNA ligase is like a glue, a super glue, right? That joins fragments together, right? Because there is a strand known as a lagging strand, right? Whereby that strand occurs in short fragments, right? In fragments. Why that is so we are going to see. So there's an enzyme that comes in and join them and join those short fragments. That enzyme is known as a DNA ligase. So these are DNA replication enzymes and what they do. You may want to read them up later. Okay, so let's look at DNA replication. There is what we know as a replication fork, right? And the replication fork is what is formed as DNA unwinds, right? So if you have your DNA unwinding this way, they form what looks like a Y shape, right? Known as a replication fork. So this here is the replication fork. Okay. So we are looking at what we said. This is what we said earlier. You have your two strands, one running from 5' to 3', the other one running from 3' to 5'. The blue strands, right? Then the red strand is the new strand that is being synthesized. You see, that green icon shows the DNA polymerase 3 that has a respective nucleotide. Now, earlier we say Okazaki fragments or lagging strand. The strand that runs from 3' to 5' is synthesized in short fragments, right? And those fragments are known as Okazaki fragments, named after the Japanese scientist Reidi Okazaki, who discovered them, right? Okay, that is replication forks. This slide talks about a replication fork. Okazaki fragments, DNA polymer, DNA replication on these strands will be looked at in the next slide. You will understand Okazaki fragment when we get to the replication slide, please. Okay. I said on this slide, we have a replication fork. The replication fork is like the Y shape that is formed as the DNA, as DNA opens up or unwinds, right? And we said that DNA replication takes place on both strands. The strand that runs from 3' to 5', and the one that runs from 5' to 3', right? So the ones that run from 5' to 3' is usually the lagging strand. Why that is so, I will explain later. This one that runs from 3' to 5' is the leading strand, right? 3' to 5'. This one runs from 5' to 3'. The one that runs from 3' to 5' is the leading strand. The one that runs from 5' to 3', replication there is known as the lagging strand. Okay. So here, DNA replication process now. on the leading strand. RNA primase enzyme begin the replication process by building a short, small complementary RNA segment known as RNA primers, about 10 to 60 nucleotide long. Then DNA polymerase 3 will begin to add the nucleotides, as we said earlier. Now, listen to this. DNA replication, okay, let me come to this slide to explain that. Now, look at this slide that I intentionally left blank. We have two strands. Of our DNA. This one runs from 3' to 5'. This one runs from 5' to 3'. These are the parental strands. They are now opened up. Now, here, the ones on 3' to 5', let's assume the original nucleotides or nucleotide sequence on it is A, T, C, G, A, T, C, G. Because they are parental strands, right? The complementary strand on the other one will be T, A, G, C, T, A, G, C. Now, when we looked at DNA structure, we said there is a 3', right? The carbon 3 of the pentyl sugar with a free hydroxy group, right? That 3' is necessary here for DNA replication because that is where the incoming nucleotide will join itself to with the carbon 5 carrying the phosphate group. We explained this also, we still mentioned today during phosphodiester bond formation, right? So this 3' is available on this end when this DNA opens, like this, right? So DNA can occur by the incoming nucleotide simply joining itself to this free 3' hydroxy group. It is simple as that. So we call this one the leading strand because there is no impediment to DNA synthesis. But this other strand, the one that runs from 5' to 3', right? DNA replication cannot start from 5' to 3', right? It can only occur from 3' to 5'. And when that occurs, okay, let's form the new strands now that will be formed here. So we have here will be T, here will be A, G, C, T, T, A, G, C. Then this strand, the second strand, okay, let's call this one the first strand, let's call this second strand. On the second strand, when DNA replication, when the DNA strands open up, like we have here, when the DNA strands open up like we have here, there is no, here, okay, let me clear this, don't worry. When the DNA strands open up, you will see that this one is 3', this is 3' to 5', right? So it is easy for replication to start from the 3' to 5' end. But this one is not exposed because the fork is like this. The replication fork is like this. Look at this slide. This slide. You will agree with me that it is not easy. There is no free prime. There's no free 3' here, but you have 3' here. So DNA synthesis can occur straight like that. But here, the 3' is not exposed for this other strand. The 3' is here. This is the 5', and this is 3'. So there is no available 3' for it to begin DNA replication. So there is the need for the we need a free 3' group. So the primer will now go and look in between along the opened length and look for a free 3'. It will see it in the middle. Because, like I said, okay, let's this is our strand. These are our strands. It has closed up here, right? So the 3' is somewhere, is not accessible. So the enzyme will now look for, on the length, on the open length, we look for free 3' that is available, right? So synthesis on that strand will now be this side, goes this way, from the 3' that is here in the middle to 5' direction now. But DNA synthesis occurs in the 5' to 3' direction. That is the new strand that you form, because they are complementary. Because the leading strand is 3' to 5'. So the DNA that you will form, the new strand, will run anti-parallel to the old strand, right? So on this lagging strand, the synthesis occurs in short fragments, because it is not easy for the DNA, it is not possible for the DNA to start at the end of the 3' that is not available, that we say it is hidden here, somewhere here. So it occurs in short fragments after some bases, right? After some nucleotides, it will cleave off and need another RNA primase to come in and make short fragments. As you can see, you have 3 fragments here, for instance. So these fragments are known as Okazaki fragments, and that is seen on the lagging strand. Now, because the leading strand, synthesis occurs directly, there is no stoppage. We say that synthesis on that strand is continuous, right? Then the lagging strand, where you have short fragments, we say synthesis there is discontinuous, because it has to start, stop, start, stop, start, stop. So we say that it is discontinuous. Then, because DNA replication can occur in both ways, that it can occur from this side. Sorry, it's becoming messed up. Now, because DNA replication can occur either ways, that is, it can go from here to here, it can also go from here to here. We said it is a bidirectional. We say it is a bidirectional. So let's go back to our slides on DNA replication and the, sorry, let me clean my drawings. Okay, so RNA primase, like I said earlier, we bring RNA primers and lay the foundation. So when that foundation is laid, DNA polymerase 3 will begin to build on it by adding a respective nucleotide. So since DNA polymerase 3 only builds in the 5' to 3' direction, so what we mean by building in 5' to 3' direction is this. I need to come back to this free slide again. We said DNA synthesis occurs from 5' to 3'. Is because when you have your strands opened up, this is the original strand, 3' to 5'. These are the original strands, the old strand or the parental strands. Now, the one that you will make will be like this, like the blue color. You will agree with me that we said that DNA is anti-parallel, that this one, the new ones in blue, if this one is 3' to 5', this one will be 5' to 3'. So that is what we meant by DNA. Then this one that is 5' to 3', the new strand that will be formed on needs to be 3' to 5'. That is what we mean by DNA replication moves or occurs in 5' to 3' direction. It is different from original 3' to 5' strand. The new ones that you have now move from 5' to 3' direction. Okay, so the two strands begin to be assembled in opposite directions, because one runs from 3' to 5', the other one from 5' to 3'. So, and we said that it's a DNA strand is bidirectional, so but it is opposite. One is going this way, like we see here. One is going this way. This arrow is going right, whereas the other one is going left. So synthesis on both strands occurs in opposite direction. And that is where we say that synthesis is a bidirectional. It's bidirectional. So no need for RNA primase to add additional primers. This is called the leading strand because DNA polymerase 3 is able to continue continuously, continue synthesis continuously. So it said that there is no stoppage, there is no breakage as seen on the lagging strand. So that is DNA replication on the leading strand. Let me take a summary of this. One, step by step, the DNA opens up by DNA helicase, right? When that occurs, RNA primase will come and lay the foundation on the 3' to 5' strand, the strand that runs 3' to 5', 3' to 5'. Then, when that occurs, after laying the foundation, DNA polymerase 3 will continue by bringing the correct nucleotide. For A, it will bring a T. For G, it will bring a C. For C, it brings G. For T, it brings A. For A, it brings T. And that synthesis goes like that, uninterrupted on the leading strand. That is the one that runs from 3' to 5'. But on the other strand, that is the other template, the other strand that runs from 5' to 3', there is no free 3' hydroxy group. Free, F-R-E-E. That is, there's no 3' hydroxy group that is free. As a result of that, consequently, DNA synthesis or replication occurs along the open strands in the middle, not at the end, because that is where you can see free 3'. You cannot see it at the end. So when that occurs, DNA synthesis on that strand will go opposite of the leading strand, right? And it occurs in short fragments because it is not easy moving opposite direction, right? So it looks, just see, okay, after short fragments, it cuts, another RNA primase will come again. But on the leading strand, primase occurs once. So if I ask you if you have 3 strands on the lagging strand, for instance, you have three strands on the lagging strand. How many RNA primase will be found? You have RNA primase coming in 3 times, but on the leading strand, RNA primase only work once. Once it lays the foundation once, it goes off. But on the lagging strand, each fragment will have the RNA primase starting them, right? So on the lagging strand, DNA replication occurs in opposite direction, moving away from the replication fork. This is the replication fork at this center that looks like a Y. It's known as the replication fork, right? This is the replication fork here. So DNA replication on the leading strand moves towards the replication fork, whereas DNA replication on the lagging strand moves away from the replication fork. Okay. Who are the people staying high J on the group? Why are you dropping IJ? So RNA primase attaches another primer, allowing DNA polymerase 3 to begin to form new point. We've explained that. The pattern created on the second strand is a series of RNA primers and short DNA fragments, known as Okazaki fragments. We have explained that. Then DNA polymerase 3, DNA polymerase 1 will remove the RNA primase after the whole process and replace them with correct DNA nucleotide, right? What we are saying there is that if you have your original template DNA strands, okay, let it be read. You have it, this is the original strand, right? And RNA primase comes and put RNA primers. So if you have A here now, if you have A here, RNA primase, what is RNA? It puts a U because there's no T in RNA. So what happens? Then DNA polymerase 3 will now come and help you to complete this strand with A, G, C. The same thing is seen here. You first have short RNA primase, then your strand will be elongated that way. You have short RNA primase, your DNA will come. You have short RNA primase, then DNA nucleotides will be joined. So at the end of the whole exercise, you have DNA polymerase 1. You can leave the class if you are tired. Oh no, marvelous, or I can kick you out. If you ask me any question, you can leave or I will kick you out. If you are tired, you can go. Okay, so after the whole process, we have an enzyme, DNA polymerase 1, that does two things. The first thing it does is that, one, it removes the RNA primers that are synthesized by primase. Secondly, it now replaces the empty nucleotide. For instance, now, if RNA primase come and removes this one is blank, the A is absent, right? A has no complementary nucleotide. So the same DNA polymerase 1 will now substitute that A, right, with a T. We now bring T to it. You know, this one was originally U. This one was originally U because it is RNA primase that brings that primer, that brings the nucleotide to it. And we said in RNA, there is no T, but a U. But after the whole process, DNA polymerase 1, now not DNA polymerase 3. DNA polymerase 1, I said, after the whole process, right? DNA polymerase 1, another enzyme. We actually have DNA polymerase 1, polymerase 2, and polymerase 3. And they do different things. DNA polymerase 3 has more nucleotides, right? But DNA polymerase 1 will come and do two things. One, it removes the RNA primers, all the U's and other RNA primers that are there, about 10 to 16 long, right? It will remove all of them and replace them with the correct DNA nucleotide. So for A, it just changes on to T. It will do that for all the small RNA primers that are formed and introduce DNA primers to this end. Then after that, these Okazaki fragments, you know, there are gaps in between them. There are gaps because they are short fragments. There are gaps. There are gaps. Another enzyme that we call DNA ligase, that we call superglue, right? We now come and remove this gap. It will close them up. It seals the nick. DNA polymerase, DNA ligase will come and join this fragment to this fragment and join this fragment to this fragment. So you now have your straight chain of a nucleotide sequence. So that is what we have during DNA replication. So DNA ligase catalyzes the formation of phosphodiester bond to seal the strand. So this gap that you are seeing here on the lagging strand will be closed up by DNA ligase. And that concludes DNA replication. That concludes DNA replication. Before we go on, do we have any question? Let's see whether we can take one on YouTube and one on Zoom. So let's go to YouTube first. Let's go to YouTube. My YouTube family, where are you? Okay. I hope you are here. Okazaki fragments. What about Okazaki fragments? I'm seeing question plenty. Okay, but you can't answer a single. You can't even ask anyone. Can RNA primase serve as a catalyst? They are all enzymes. What does enzymes do? Enzymes are catalysts now. They are catalysts. All these enzymes that you see, they are catalysts because they catalyze a reaction. RNA primase, for instance, is a catalyst in that regard. So I really don't, what is this? I really don't I don't understand DNA replication process. Your explanation on the opening of 5' was not clear. I don't understand slide 41. Okay, I've had enough. Let's go to Zoom. Slide 41, DNA replication. I didn't say much on DNA replication. I only said DNA replication is a process whereby, okay, it's DNA replication that we are treating here. Okay, somebody says slide 41. Let's see. Can you recap very fast? I'm confused. This part of replication. Slide 41. I don't understand the DNA polymerase 3 and polymerase 1. Okay, slide 41. I was on slide 41. There's nothing to explain on slide 41. Please go and read up this. They are just like I tell you, you are driving your car. Your brake is to stop. That is what is happening here. Your throttle accelerator is to move. That is what is happening on this slide 41. These enzymes do certain things in the process of DNA replication, and their functions are listed. So I don't think I need to read DNA primase. It synthesizes RNA primers. Helicase unwinds parental the double helix at replication forks. So this is the enzymes and what they do. I don't think there's anything to explain for that here. But okay, let me take them again. DNA helicase, like you have your DNA double strands, like your zip, right? That's I don't know the name of that thing that's used to unzip your zip. That when you drag it down, it opens. When you drag it up, it closes. That can be likened to your DNA helicase. It helps to unwind the DNA. Then when that DNA unwinds, there are enzymes, or there's an enzyme that prevents it from closing up again because there's tendency for them to close up, to reanneal, to come together. But once that enzyme binds to it, it holds it apart. It holds them apart. That enzyme is known as the single strand binding proteins. The single strand binding proteins bind on the open strands so that they don't have the ability to join. Just like once it binds to it, it stops them from closing up. Then DNA gyrase or topoisomerases prevents knots from forming or torsion from forming as the DNA opens up. You know, there's tendency just like, well, you have your rope, two ropes, one around each other. You just want to open them vim at a time. It's possible that at once, it's possible that it forms knots, then it stops moving, right? That enzyme that prevents that from occurring is a DNA gyrase or topoisomerases. Primase lays the foundation by bringing RNA primers. DNA polymerase 3 adds the nucleotides. DNA polymerase 1 removes RNA nucleotides and replaces them with DNA nucleotides. The DNA ligase superglue that joins the short Okazaki fragments together. Please, let's move on. We still have a long way to go, a very long way to go. Then slide 44. Somebody mentioned slide 44 is just showing the process of DNA replication. The leading strand and the lagging strand. I said on the leading strand, that is the strand that synthesis there starts from the parental strand that runs from 3' to 5'. And I referred you back to our class on phosphodiester bond, this slide. Okay, let me go back to that slide. Okay, this slide. The 3' and the 5'. So for DNA replication to occur, right, this has to be free. This position needs to be free. This free prime, this free prime, this 3' has to be free so that the incoming nucleotide can bind to it with its phosphate carrying carbon 5, right? So that is what we are seeing again. And I remember saying that there is 5' to 3'. So this 3' is very, very important. It's very, very important in such a way that it needs to, like you can see the next slide here, the next nucleotide here, the 3' is free for an incoming nucleotide to join. So that 3' needs to be, there is the need for it to be available so that an incoming nucleotide can attach to it. And that knowledge is what we transfer here to slide 44. To slide 44 here. This 3' is talking about that 3' on that your pentose sugar that opens up, it is free so that an incoming nucleotide can bind to it with 5' that is carrying a phosphate group to form a phosphodiester bond. So synthesis starts from there and goes in 5' to 3' direction. But on the lagging strand now, on the lagging strand, there is no free 3'. So synthesis starts from the middle and it occurs in short fragments. And it goes opposite direction of the leading strand, and it moves away from the replication fork. Some people said they have class, and we are still going to slide 71 or 72, and some people are, please, okay, somebody asked which class some people are leaving already. Please, which class is that? Which class do you have, Tumishe? Which class do you have? PHS 103. PHY 103, PHY 103. Okay, those are okay. Please, you can join your class. We will start from here next time. That means we need to move very fast because I must conclude my class with you on Friday. So we have a long way to go on Friday. Thank you. Bye-bye, everybody. Okay, if you don't have any class and you have question or clarification, you can stay. You can stay so that I can answer your question, but I won't go further. I will stop here. So if you have any question, you can ask while others join their class. So those of you that have clarifications, we can take it. If there is no question, we can share the grace. Meaning of prime numbers on the carbon. Okay, that means we were not in our last class. We said the pentose sugar has carbon. Nucleotide, the nitrogenous bases also have carbon atoms. So to distinguish one carbon from the other, to distinguish the carbon of the ribose sugar from the carbon of the nitrogenous bases, we put prime on the carbon of the sugar. So that is why you have prime on that carbon of sugar. The leading strand, is it 5' to 3'? No, the leading strand is the original strand or the parental strand that runs from 5' to 3'. That is the lagging strand. The leading strand is the one that runs. Let me see your question again. The leading strand, is it constant for 3 prime. Yes, definitely. Okay. The leading strand, is it constant? Is it constant for 3 prime? Yes. The leading strand also always runs from 3 prime to 5 prime. Then the lagging strand always run from 5 prime to 3 prime. Experiment on semi-conservative method of replication. I've given you that as an assignment, so I will not dwell further on that. I said go and write in details that experiment. The class is for pre-med. We can go on. Are they not doing biology? The Okazaki fragments also called lagging strand, or they are parts. The Okazaki fragments are found on the lagging strand. They are not called the lagging strand. Because they are the lagging strand because the lag, the way they form, it's like, as the word implies, lag. When something is slow, right, it doesn't follow the other one, right? So the new strand that is synthesized is the lagging strand, and they use Okazaki fragments on the lagging strand. Chaga's rule again. Wait. Okay, thank you, everybody. Take care and enjoy the rest of the day. Please go and join your classes or go and do another thing. Please, we may have to move very fast on Friday because we do 8 to 10.